to be activated. And why would a gene be activated? To make the proteins for the cell that needs to do the behavior. So the truth statement is this. When a gene product is needed, a signal from its environment, not an emergent property of the gene itself, activates expression of the gene. Well, that's somewhat of a complicated sentence, so let's simplify it. Just read line 2 and line 4. And if we read that, it says, a signal from its environment activates expression of the gene. What does that mean? The genes in your body are selected not because they're self-selecting. The genes are always selected in response to the environment that you're in. So if you had that cancer gene and for 35 years, let's say, you stood around saying, hey, I don't have cancer, and all of a sudden cancer happened, are we going to go to the gene and blame the gene, or what would we actually look for as responsible if you understand the truth statement? The signal from the environment. What change in your life promoted activation of the gene that was sitting dormant for 35 years? We've been focusing on the gene all the time. The point is, we have to start focusing on the signals. The signals do this. Well, let me explain how this happens. When we look at a cell nucleus, this is where the chromosomes and the DNA is. I can stain the chromosomes, and you break open the nucleus, you can see all these different chromosomes. You have 23 pairs of chromosomes to make a human. They're called pairs of chromosomes because you get 23 chromosomes from your mother, 23 chromosomes from your father, and they're matched pairs. So, in fact, even I can see that one of these is from the mother and one of these is from the father because you can color code them. Well, it's a nice, interesting experiment, but the question is this: what am I staining? I'm not staining DNA. So what's in the nucleus? And the answer is this. 50% of the nucleus is DNA and 50% is protein. And the reason why we have a problem here is this. For 50 years, everyone was so focusing on the genes that when they wanted to study the DNA, what did they do? They'd go find a nucleus from the cell, they'd break it open, expose all the chromosomes, and you know what they do? Separate the protein from the DNA and then throw away the protein. And for 50 years, they've thrown away the protein in their focus on studying DNA. And now, all of a sudden, in the last few years, the question is, hey, what have we been throwing away? And the answer is the control. For 50 years, they've thrown the control away, what controls the genes, and studied pure DNA. There is no such thing as pure DNA in any organism. The DNA is always associated with the protein. So what's the function of the protein? Look how simple this is. The protein forms a sleeve around the DNA. So let me give you an example of what I mean by that. Imagine my bare arm is a gene. Let's say it's a gene for blue eyes. And I say, okay, can you read the gene for blue eyes, yes or no? Yes. Okay, but all of a sudden, well, what does the DNA look like when I put it back in the nucleus? Put the sleeve of protein on it. Can you read the gene for blue eyes, yes or no? If you want to read the gene for blue eyes, what do you have to do? Take the sleeve off. Well, the sleeve is protein. How does a sleeve come off? Here's the protein, and it's locked onto my arm. If you can remember back about 15 minutes ago, what is it that will cause the change in the shape of the protein? So when I add the signal from the environment, all of a sudden, then what happens is this. The protein changes shape, pulls away from the DNA. Now I can read the gene, and when the signal is removed, the protein will come back and cover up the sleeve again. So the bottom line was this. The gene was just sitting there all the time. It's whether the proteins are present or absent. So if I look at it this way, then we understand this. I said you were made out of protein. The understanding is that DNA is the blueprint for the protein. In conventional textbooks, because they've thrown away the protein for 50 years, they don't talk about this. Conventional talks about the DNA goes to the RNA, which is like a Xerox copy of the DNA, and the RNA is then turned into the protein. And then they talk about the primacy of DNA. That's what's in all the textbooks. You are a result of your DNA. But they've thrown away the protein. So when we put it back in, it says, the protein covers up the DNA. The protein is a sleeve. But to take the sleeve off, you actually have to have the environmental signal. So remember what Niehaus' quote was? A signal from the environment activates the expression of the DNA. So the bottom line right here is this. The environmental signal comes in and changes the shape of the regulatory protein, which removes the sleeve, exposes the DNA, and then I can make my proteins. So rather than the primacy of DNA, which is conventional thought, it's actually the primacy of the environment. It's the environment that selects your genes, not the genes themselves. So if I wanted to illustrate it, let's go back to our picture of how the cell worked. What I showed you was this. The signal from the environment activated the receptor, which activated the effector, and the effector activated the secondary signal to go down to the protein. Remember that picture just a minute ago? Well, here's the point. In this illustration, the protein's not there. And if I need the protein because the environmental signal, I have to respond to the signal, and the protein's not there, what would I need to do if the protein's not present in the cell? Go to the nucleus and activate the gene for the making of the protein, right? So let's watch the behavior of this as it happens. So basically what's going to happen is this. The environmental signal joins to the receptor, activates this whole process so that I activate this, but look, the signal goes down, the proteins are missing. If the proteins are missing, I need the proteins to make the proper response, but they're not there. So what I have to do then is take this signal and go into the nucleus of the cell where the DNA is. But the DNA is covered up by a sleeve of protein. And at periodic points, at every gene, there's a control protein called a regulatory protein. And you know what happens? The signal from the environment binds to the right gene by the shape. It doesn't bind to this one, wrong shape. Binds to this one. Now, what happens when a signal binds to a protein? Changes the shape of the protein. And watch what happens. As soon as I change the shape of the protein, I cause the sleeve to come off the DNA. And when it happens, look what I'm exposing. The gene is now exposed. And what am I going to do with this? Well, I need to make a copy of the gene called RNA, which then goes into the cell where it's turned into the protein. So the bottom line is, then I take the RNA molecule, make a copy of this DNA molecule, and then this is a blueprint of the gene, and this is called RNA, messenger RNA, and this blueprint is actually used to make the protein. So what's the understanding about this whole process? It says this, the gene is not exposed until the signal calls it into play. When the signal is gone, the sleeve covers it up, and the gene is now hidden. If this is a cancer gene, and it's not giving you cancer because it's not exposed, what will cause the cancer gene to express itself? The signal. So all of a sudden it says, then what signals from your environment are you perceiving that you are selecting negative genes or processes in your body? And all of a sudden we start to see, so let's do the same thing now without all the labels, real fast. The secondary signal goes down, the protein's missing. The secondary signal goes into the nucleus, finds the right gene by binding to the right regulatory protein, causing the regulatory protein sleeve to come off, exposing the gene. And once the gene's exposed, I make a copy of it called RNA, and this is what goes out into the cell and is used for the function and behavior of the cell. So what is the conclusion of all of this? And the answer is simply this. Perception controls genes. Understand? The genes did not control themselves. The perception was the signal that converted the sleeve of the DNA to come off. So the bottom line is, perception not only controls behavior, but perception will actually go in and select which genes you're going to express. Now here comes the third part. The first part, perception controlled behavior because the protein was there. The second part, the signal shows up and the protein's not there, so the perception signal goes to the genes and activates the appropriate gene. Here's the third part. What happens if I run into a stressful environment and I don't have the appropriate genes to respond to that stress? Well, then you just have to say, well, the only way you're going to manage is to change the genes. Well, in conventional biology, the only way the genes change is a process called random mutation. This is in all the textbooks. What does it mean? It says this. I can chemically cause a mutation to occur, but what I can't control is the outcome. The outcome is always random. So the bottom line is that's where Darwinian belief comes in, that evolution was random changes in the genes, that genes are only changing by accident. That's conventional belief. Except 1988, this paper comes out in Nature by a man called John Cairns. And it changes the entire foundation of biology that we've ever held for the following reason. he tells us about a new kind of mutation called an adaptive mutation. The point about this mutation? That the genes are not changing randomly, but the environment is controlling the mutation. So that you're always adjusting your genes to